Hello dear students, I'm your instructor Abid Jan. This is lecture number 22 which will cover DNA replication in prokaryotes from the course of molecular biology. Learning objectives. By the end of this section, you will be able to do the following. Explain the process of DNA replication in prokaryotes and discuss the role of different enzymes and proteins in supporting this process. The process of replication. DNA replication has been well studied in prokaryotes primarily because of the small size of the genome and because of the large variety of mutants that are available. E. coli has 4.6 million base pairs in a single circular chromosome, and all of it gets replicated in approximately 42 minutes, starting from a single site along the chromosome and proceeding around the circle in both directions, bidirectional replication. This means that approximately 1,000 nucleotides are added in 1 second. Thus, the process is quite rapid and occurs without many mistakes. DNA replication employs a large number of structural proteins and enzymes, each of which plays a critical role during the process. The key players. One of the key players is the enzyme DNA polymerase, also known as DNA pol, which adds nucleotide one by one to the growing DNA chain that is complementary to the template strand. The addition of nucleotides requires energy. This energy is obtained from the nucleotide triphosphates ATP, GTP, TTP, and CTP. Like ATP, the other NTPs, nucleotide triphosphates are high-energy molecules that can serve both as the source of DNA nucleotides and the source of energy to drive the polymerization. When the bond between the phosphate is broken, the energy released is used to form the phosphodiester bond between the incoming nucleotide and the growing chain. In prokaryotes, three main types of polymerases are known: DNA pol 3, DNA pol 1, and DNA pol 2. It is known that DNA pol 3 is the enzyme required for DNA synthesis. DNA pol 1 is an important accessory enzyme in DNA replication, along with DNA pol 2, and are primarily required for repair during replication. How does the replication machinery know where to begin? It turns out that there are specific nucleotide sequences called origins of replication, where replication begins. In E. coli, which has a single origin of replication on its one chromosome, as in most prokaryotes, this origin of replication is approximately 245 base pairs long and is rich in the AT sequences. The origin of replication is recognized by certain proteins that bind to this site. An enzyme called helicase unwinds the DNA by breaking the hydrogen bonds between the nitrogenous base pairs. ATP hydrolysis is required for this process. As the DNA opens up, Y-shaped structure called replication forks are formed. Two replication forks are formed at the origin of replication, and these get extended bidirectionally as replication proceeds. Single-strand binding proteins coat the single strands of DNA near the replication fork to prevent the single-stranded DNA from winding back into a double helix. DNA polymerase has two important restrictions. It is able to add nucleotides only in the 5' to 3' direction. That is, a new DNA strand can be only extended in this direction, 5' to 3'. As you can see here, this is 5' to 3'. This is 5' to 3'. This is 5' to 3', and similarly this is 5' to 3'. It also requires a free 3' hydroxyl group to which it can add nucleotides by forming a phosphodiester bond between the 3' hydroxyl end and the 5' phosphate of the next nucleotide. This essentially means that it cannot add nucleotide if a free 3' hydroxyl group is not available. Now, the question is that how does it add the first nucleotide? The problem is solved with the help of a primer that provides the free 3' hydroxyl end. This is the primer in orange color, and this provides the free 3' hydroxyl group, and then it can be extended by the polymerase. Another enzyme, RNA primase, this one sea green color, synthesizes an RNA segment orange color, that is about 5 to 10 nucleotides long and complementary to the template DNA. Template DNA is in red color. Because this sequence primes the DNA synthesis, it is appropriately called the primer. DNA polymerase can now extend this primer in 5' to 3' direction, adding nucleotides one by one that are complementary to the template strand. Visual connection. A replication fork is formed when helicase, this enzyme, separates the DNA strand at the origin of replication. The DNA tends to become more highly coiled ahead of the replication fork. This is called topoisomerase. This enzyme, this green ring, breaks and reforms DNA's phosphate backbone ahead of the replication fork, thereby relieving the pressure that results from this supercoiling. Single-strand binding proteins, oval shape, purple color, bind to the single-stranded DNA to prevent the helix from reforming. Another enzyme, PROKARYOTES, this one, synthesizes an RNA primer, this orange color primer. The DNA polymerase 3, this one, uses this primer, this RNA primer, to synthesize the daughter DNA strand. On the leading strand, this is the leading strand. On the leading strand, DNA is synthesized continuously. This is continuous synthesis from 5' to 3'. Whereas on the lagging strand, DNA is synthesized in short stretches called Okazaki fragment. This is one fragment, 5' to 3'. This is 5' to 3'. This is 5' to 3', and this is 5' to 3'. So these are short stretches. This is one stretch, this is another, this is another, and this is another. And then DNA polymerase 1, the green in color, replaces the RNA nucleotides with DNA nucleotides. The DNA ligase, this one, seals the gaps between the Okazaki fragments, joining the fragments into a single DNA molecule. The replication fork moves at the rate of 1000 nucleotides per second in a typical prokaryotic cell. Topoisomerases prevents the overwinding of the DNA double helix ahead of the replication fork as the DNA is opening up. It does so by causing temporary nicks in the DNA helix and then resealing it. Because DNA polymerase can only extend in the 5' to 3' direction, and because the DNA double helix is anti-parallel, there is a slight problem at the replication fork. The two template DNA strands have opposing orientation. This is one template and this is another template, and if you see, this is 5' to 3', and this is 3' to 5'. So this strand is reverse in orientation with respect to this strand. One strand is in the 5' to 3' direction, and the other is oriented in the 3' to the 5' direction. Only one new DNA strand, the one that is complementary to the 3' to the 5' parental DNA strand, can be synthesized continuously towards the replication fork. This continuously synthesized strand, this one, is known as leading strand. The other strand, this one, complementary to the 5' to 3' parental DNA, is extended away from the replication fork. Now, this synthesis is occurring in this direction. This is the replication fork. Leading strand is approaching towards the replication fork, and lagging strand is moving away from the replication fork. And the synthesis of this lagging strand occur in fragments known as Okazaki fragments, each requiring a primer to start the synthesis. New primer segments are laid down in the direction of the replication fork, but each pointing away from it. Okazaki fragments are named after the Japanese scientist who first discovered them, and the strand with the Okazaki fragment is known as lagging strand. The leading strand, this one, can be extended from a single primer, whereas the lagging strand needs a new primer for each of the short Okazaki fragments. The overall direction of the lagging strand will be 3' to 5', and that of the leading strand is 5' to 3'. Another protein called the sliding clamp this one, this is a sliding clamp, and this is the DNA polymerase 3. Same is the case here. This is the DNA polymerase 3, and this is the sliding clamp. holds the DNA polymerase in place as it continues to add nucleotides. The sliding clamp is a ring-shaped protein that binds to the DNA and holds the polymerase in place. As synthesis proceeds, the RNA primers are replaced by DNA. The primers are removed by the exonuclease activity of DNA polymerase I, which uses DNA behind the RNA as its own primer and fills the gaps left by removal of RNA nucleotides by the addition of DNA nucleotides. The nicks, there is a nick, similarly we have here that remain between the newly synthesized DNA and the previously synthesized DNA are sealed by the enzyme DNA ligase, which catalyzes the formation of phosphodiester linkages between the 3' hydroxyl end of one nucleotide and the 5' phosphate end of the other fragment. Once the chromosome has been completely replicated, the two DNA copies move into two different cells during cell division. The process of DNA replication can be summarized as follows. DNA unwinds at the origin of replication. Enzyme helicase opens up the DNA forming replication forks. These are extended bidirectionally. Single-strand binding proteins coat the DNA around the replication fork to prevent rewinding of the DNA double helix. Enzyme topoisomerase binds at the region ahead of the replication fork to prevent supercoiling. Enzyme primase synthesizes RNA primers complementary to the DNA strand. The main replicating enzyme, DNA polymerase III, starts adding nucleotides to the 3' hydroxyl end of the primer. Elongation of both the lagging and the leading strand continues. RNA primers are removed by exonuclease activity of DNA polymerase. Gaps are filled by DNA polymerase I by adding DNA nucleotides, DNTPs. And finally, the gap between the two DNA fragments is sealed by the enzyme DNA ligase, which helps in formation of phosphodiester bonds. The table summarizes the enzymes involved in prokaryotic DNA replication and the function of each. DNA polymerase 1 removes RNA primers and replaces it with DNA nucleotides. DNA polymerase 3, being the main replicating enzyme, adds nucleotide in the 5' to 3' direction. Helicase opens up the DNA double helix by breaking hydrogen bonds between the two strands. DNA ligase seals the gaps between the Okazaki fragments to create one continuous DNA strand. The enzyme primase synthesizes RNA primers needed to start the process. The sliding clamp helps to hold the DNA polymerase in place when nucleotides are being added, and the topoisomerase helps to relieve the strain on DNA when unwinding by causing breaks and then resealing the DNA. And SSB, single-strand binding proteins, binds to single-stranded DNA to prevent DNA from rewinding back into the double helix. Link to learning. You can review the full process of DNA replication by pointing your browser to the URL. Section Summary. Replication in prokaryotes starts from a sequence found on the chromosome called the origin of replication, the point at which the DNA opens up. Enzyme helicase opens up the DNA double helix, resulting in the formation of the replication fork. Single-strand binding proteins bind to the single-stranded DNA near the replication fork to keep the fork open. Enzyme primase synthesizes an RNA primer to initiate synthesis by DNA polymerase, which can add nucleotides only to the 3' end of a previously synthesized primer strand. Both new DNA strands grow according to their respective 5' to 3' directions. One strand is synthesized continuously in the direction of the replication fork. This is called the leading strand. The other strand is synthesized in a direction away from the replication fork in short stretches of DNA known as Okazaki fragments. This strand is known as the lagging strand. Once the replication is completed, the RNA primers are replaced by DNA nucleotides and the DNA is sealed with DNA ligase, which creates phosphodiester bonds between the 3' hydroxyl of one end and the 5' phosphate of the other strand. Visual connection questions. You isolate a cell strain in which the joining of Okazaki fragments is impaired and suspect that a mutation has occurred in an enzyme found at the replication fork. Which enzyme is most likely to be mutated? Once again, I will repeat the question because this is a visual connection question and you need to focus on this figure. You isolate a cell strain in which the joining of Okazaki fragments is impaired. This is one Okazaki fragment, this is another, and this is another. Now, this is the joining point, this is the joining point, this is the joining point. Now, which enzyme is most likely to be mutated if this joining of two fragments is impaired? Of course, it's the DNA ligase, as this enzyme joins together Okazaki fragments. Critical thinking questions. Number 1. DNA replication is bidirectional and discontinuous. Explain your understanding of those concepts. At an origin of replication, two replication forks are formed that are extended in two directions. On the lagging strand, Okazaki fragments are formed in a discontinuous manner. The next. What are Okazaki fragments and how they are formed? Short DNA fragments are formed on the lagging strand, synthesized in a direction away from the replication fork. These are synthesized by DNA polymerase. Next, if the rate of replication in a particular prokaryote is 900 nucleotides per second, how long would it take 1.2 million base pair genomes to make two copies? So the answer is around 22 minutes. Next, explain the events taking place at the replication fork. If the gene for helicase is mutated, what part of replication will be affected? At the replication fork, the events taking place are helicase action, binding of single-strand binding proteins, primer synthesis, and synthesis of new strands. If there is a mutated helicase gene, the replication fork will not be extended at all. What is the role of a primer in DNA replication? And what would happen if you forgot to add a primer in a tube containing the reaction mixture for a DNA sequencing reaction? As you know, primer provides a 3' hydroxyl group for DNA polymerase to start adding nucleotides. There would be no reaction in the tube without a primer, and no bands would be visible on the electrophoresis. Quinolone antibiotics treat bacterial infections by blocking the activity of topoisomerases. Why does this treatment work? Explain what occurs at the molecular level. Prokaryotes bacteria treated with quinolones will no longer be able to replicate their DNA, and topoisomerase relieves the excess DNA supercoiling that occurs ahead of the replication fork as DNA is unwound for replication. If topoisomerase is inhibited, DNA helicase will only be able to unwind the DNA for a short stretch before the supercoiling becomes too overwound for replication to continue. The bibliographic information. Stay curious and all the best for your life ahead.